Hello everyone, now we are learning about the nitrosamine impurities in human drugs. Guidance taken to make this presentation is Control of Nitrosamine Impurities in Human Drugs, published by US FDA on February 2021. Current revision is 01. What is nitrosamine? Nitrosamines are class of compounds having the chemical structure of nitroso group bonded to an amine. In the below image we are seeing amine is bonded to the nitroso group and forming the nitrosamines. Why nitrosamines are controlled? Nitrosamines are probable human carcinogen, cancer causing agent. Due to this reason, pharmaceutical companies must control these impurities in the drug product and drug substance. How the nitrosamines are forming? Nitrosating reaction between the amines and the nitrous acid on forming the nitrosamines. And here another two examples are given. Sodium nitrate reacts with amine under acidic condition and forming the N-nitrosoamine. Dimethyl formide undergoing minor decomposition and forming the dimethylamine. Further, under condition of sodium nitrate and nitrous acid, forming the NMDA, N-nitrosodimethylamine. As per the FDA guidance, total 7 nitrosamine impurities are identified. Those are NDMA, NDEA, NDBA, NMPA, NDIPA, NIPEA, NMBA. These are the structures given in the US FDA guidance for the nitrosamine impurities. And the root causes for the nitrosamine generation are secondary, tertiary, quaternary amines, contamination in vendor sourced raw materials, recovered solvents, catalysts and reagents, lack of process optimization and control. Details are provided in the next slides. Secondary amine, tertiary amines, quaternary amines available in the API, API degradants, intermediates, starting materials, and in some cases added intentionally as reagents and catalysts. And contamination in vendor sourced raw materials through fresh solvent contamination. Sodium nitrate is a non-impurity. Some raw materials contain amine groups. Starting material or outsourced intermediates may be at risk through cross-contamination. And the recovered solvents, catalysts and reagents pose a risk of nitrosamine impurities. Lack of process optimization and control. Reaction conditions such as temperature, pH or the sequence of the adding reagents, intermediate solvents are inappropriate or poorly controlled. So these are the risk factors for the nitrosamine generation such as aliphatic, aromatic, free bases salt, API, degradant, contaminate from the solvents, intermediates, reagents, catalyst, and nitrosating agents such as reagents, nitrates, nitric acid, nitroso-alkyls, inorganic nitrate available in the excipient, nitrate in water, and conductive conditions such as pH, temperature, nitration catalyst, concentration, water presence, kinetic energies. So these all are the risk factors for the nitrosamine generation. So as per the FDA guidance, acceptable intake limit mentioned below 96 nanogram per day for the NDMA and NMBA. Rest all other impurities acceptable intake is 26.5 nanogram per day. And the risk mitigation measures proposed for the API and drug product manufacturer given below. Initially, we need to conduct risk assessment for the marketed products under approved pending application. If the risk is identified, then we have to go for the conformity testing for the presence of nitrosamine impurities. Further, we have to report these changes to the regulatory agencies. This is about the nitrosamine impurities. Thank you.